Hello, my name is Shelby Sammartino. Welcome to my talk on the vaginal microbiome sampling in bonobos or Pampeniscus. A brief background on the vaginal microbiome tells us that its composition is related to specific health outcomes. In humans, research looking at the relationship between the vaginal microbiome and behavior focuses on the abundance of the genus Lactobacillus, as it is related to the risk of sexually transmitted diseases and other infections like bacterial vaginosis or BV. BV is characterized by high vaginal pH, high microbial diversity, and an increased risk to other STDs. It's also been shown that BV is more common in women exhibiting more sexual fluidity, that is, women who have sex with both males and females. Thus, since bonobos also exhibit similar sexual fluidity and are also genetically similar to us, it's of interest to further investigate how these sexual patterns are contributing to microbial composition and pH in a non-human model. So for this preliminary study, I wanted to examine the influence of social and sexual behaviors on bonobo vaginal pH. I collected my data at Ape Cognition and Conservation Initiative in Des Moines, Iowa over a 5-week period this past winter. 3 female bonobos were trained to present their swellings for swab collection, as can be seen in the photo in the methods section, in order to collect pH and microbial samples from the vagina. I utilized pH data collected from other animal species compiled by Miller et al. To examine where bonobos fall compared to other primates and humans. We found that bonobo vaginal pH is more similar to the average human woman vaginal pH than the average non-human primate pH, as can be seen in Figure 2. The bonobo data point is circled on this chart. This is an interesting finding given the similarity of sexual patterns exhibited by both these species. Next, I will be utilizing the microbial samples I collected to further investigate the relationship between sexual behaviors, pH, and the vaginal microbiome to assess the possible health implications of sexual fluidity. The results of these studies are important for providing a more complete understanding of how social and environmental factors impact well-being and could also garner understanding about the differences in disease susceptibility across demographics. Thank you for listening to my talk. Please direct any questions you may have to shelby.sammartino@utsa.edu. Thank you.